Good afternoon. It's my pleasure to welcome you to the Florence Mahoney Lecture and to introduce you to Florence Mahoney's history, as well as to introduce you to the introducer of our speaker for today. For those of you who are not aware, Florence Mahoney was a true champion of NIH in general and of NIA in particular. Until her death in 2002 at the age of 103, she was a very avid and serious aficionado and student of science, particular basic science. I would visit her at least annually and be thoroughly grilled on the quality of science we were supporting. She was very critically involved in the selection of speakers, and I can only imagine she couldn't be more thrilled than with our speaker today, Carol Greider. In her history, her home in Georgetown and its dining room or salon was famous for the meetings she had with policymakers of the time, responsible for garnering great support for NIH overall, for the creation of NIA, as well as serving a number of other institutes. She was a remarkable woman, and oh, that we only had her with us now when we truly need her. But again, she could be no happier than with the introductions about to come. So without further ado, let me introduce Francis Collins, who will introduce you to our speaker of the day.
Thank you, Richard, and good afternoon. It's great to see the room completely packed with people who've come to hear Carol Greider's presentation, and hello also to those in Lipset in the overflow room, because I know we filled up this space. And that's appropriate, because our speaker for the Florence Mahoney Lecture on Aging is a remarkable scientist, a role model for many, and a winner of the Nobel Prize. Her past training experiences are outlined in your little program, and I won't highlight very much except to point out a couple of things that I thought were of note. Carol has been very open about the fact that she suffered from dyslexia and therefore had some significant issues early on in her formal education, but obviously rose above those in a remarkably successful way. And I know many scientists who also have that condition will resonate with what the challenges are of trying to work in a world where words are particularly taken with great importance. And obviously the kind of achievements that Carol has made should be an encouragement to all. But it should also be an encouragement in many other ways. She got her undergraduate degree at UC Santa Barbara and went on to Berkeley, where she worked quite notoriously in terms of the output of the enterprise with Liz Blackburn. And it was there that she was successful in cloning, in purifying the enzyme telomerase from tetrahymena. She went on then to do a postdoc of a very unusual sort, and I want to mention this because of the fact that we at NIH have just started a new program called the Early Independence Program, which aims to give particularly talented, gifted PhD students the chance to conduct research in a relatively independent way, with good mentoring, but in a way that they can define their own research projects and have their own resources. And that's what Carol did at Cold Spring Harbor, in one of the early examples of this kind of program, and obviously with great success, because it was there that she purified the RNA component of telomerase, a very critical next step in understanding this critical enzyme. She is now, as you probably know from looking at the program, the director, which is the Hopkins word for chairman, of the Department of Molecular Biology and Genetics, and the Daniel Nathans professor, Dan Nathans being somebody that all of us who knew him hold in great regard, and I know Carol as well takes that particular chair with great seriousness, and also professor of oncology. She's received many recognitions for her work, including the Lasker Award in 2006, election to the National Academy back in 2003, and of course most prominently the Nobel Prize in Physiology or Medicine in 2009, awarded jointly to her, to Liz Blackburn, and to Jack Szostak. And so I think, if I'm right, it would have been just about a year ago that Carol would have been in Stockholm, surrounded by a great deal of pomp, circumstance, fine food, and great speeches. And I'm sure that was a great experience, and I hope that being here at NIH in the Mazur Auditorium it compares in some slightly positive way with that, although I wouldn't really expect her to endorse that statement. Just the same, Carol, we are delighted to welcome you here this afternoon to give this lecture entitled Telomeres and Telomerase in Cancer and Age-Related Disease. Please welcome Professor Carol Greider.
Well, thank you. It's a real pleasure to be here to give the Florence Mahoney lecture. She was clearly a really wonderful supporter of basic science, so it's an honor to give this lecture in her honor. And also, before I start, I wanted to say it's very appropriate for me to be giving this as part of the National Institute on Aging, because the majority of the story that you're going to hear has really been funded over the years almost entirely by the NIA. And so I want to thank the NIA and especially Anna McCormick, who very early on, 20 years ago, got interested in what we were talking about and took me as a young scientist through the process of getting grants and how to advocate for yourself. So that really made a difference in my being able to do these kinds of experiments that I'm going to talk to you about today. So what we're going to talk about is telomeres. Telomeres are the ends of chromosomes, and they provide two really important functions. They have to protect the chromosome end, and they protect the chromosome end from any kind of degradation that would degrade the chromosome end. They protect the chromosome ends from recombination and from end-to-end fusion. Telomeres also have to be able to maintain the length of the chromosome, and we'll be spending a fair amount of time discussing this issue of length maintenance. So telomeres were first defined by H.J. Muller and by Barbara McClintock. Muller working in Drosophila, looking at the effects of terminal deletions in irradiated Drosophila, and McClintock working in corn or maize, looking at what happens when dicentric chromosomes break. And both of them came to the conclusion that there must be something special about the very end of the chromosome, and so they named this the telomere for the end part of the chromosome and said it must provide a very important function. Now, telomeres were molecularly defined first in the ciliated protozoan tetrahymena, shown here, which actually both telomeres and telomerase were identified in tetrahymena. Tetrahymena is a single-celled organism with over 40,000 chromosomes. So if you want to really understand something at a basic level, you go to the source. So Liz Blackburn and Joe Gall in 1978 first defined the sequences which were on these tetrahymena telomeres, and it turned out that that repetitive sequence, in the case of tetrahymena, it's T2G4, and in many other organisms, it's a very similar repeat. You'll be hearing about the mammalian telomeric repeat, which is T2AG3, so a very similar, simple tandem repeated sequence. So one of the things that we knew early on is that the way that chromosomes are normally replicated, the DNA polymerase can't go completely to the molecular end. So if this is the chromosome, and of course all of the genes and various things are in there, all of you omics people, what you do is all in here. And we're going to focus, however, on the chromosome ends, or the telomeres, where there are these tandem repeats. And every time the cell divides, there's a little bit of loss of the sequence from the ends of the chromosome. And this is because the replication machinery doesn't completely replicate to the very end. And so if you imagine the cells continuing to divide, you can't have an organism continuing to exist if you're going to have such erosion from the ends. And so it was apparent many years ago that there must be some mechanism to overcome this end replication problem. And that's what I worked on when I was working with Liz Blackburn. And what we found was that there is this very unusual enzyme that we named telomerase, which allows the maintenance of these chromosome ends. So although you may have some shortening due to end replication, the telomerase enzyme has within it a region which is complementary to the telomeric sequences, so that you can use this RNA template to add more telomeric sequences back on, and you can have elongation of the telomere. Now, we'll be talking about the different components of telomerase a little bit. It turns out that this very unusual enzyme contains both an essential RNA and an essential protein component. So the protein component, which is depicted here, which surrounds this region of the RNA that provides the template, the protein component we call TERT, for telomerase reverse transcriptase, because it has homology and catalytic similarity to reverse transcriptase enzymes. The RNA component we call, imaginatively, telomerase RNA. And it has a very important structure within the RNA. It's not simply a passive template. And in fact, there are regions of this RNA where you can take and make a single base pair change and eliminate catalytic activity. Again, the RNA is an essential component of the enzyme, not simply serving passively as a template. So what telomerase does then is to solve this end replication problem, to allow the balancing of telomere shortening during normal cell division. All right, so then the question becomes, why does telomerase matter? Why have we been able to study this enzyme over the last 20 years and continually find out new things? Well, it's turned out that work from many labs over the last 20 years has clearly defined that telomerase is absolutely essential for cells that have to divide many times. In cancer, if you can imagine that there's a particular tissue and that one of those cells undergoes a genetic change which would lead toward it becoming a cancer cell, that particular cell is going to divide many, many more times relative to the other cells around it. And it turns out that these cells which have become cancer cells require telomerase to solve this end replication problem. That if you can't maintain the telomeres during these large number of cell divisions, then that tumor cell will not be able to progress. A second area where telomerase is clearly essential has to do with tissue renewal. If you think about tissue-specific stem cells, you have a stem cell that has to self-renew, but then the pluripotent cells downstream have to go through many rounds of cell division in order to generate the final differentiated tissue. And it turns out that telomerase is essential in this process of tissue renewal, and we'll be talking mostly about this today. So we got interested in telomeres and cellular senescence about 20 years ago. In a collaboration with Cal Harley, we were interested in what happens in primary human fibroblasts when the fibroblasts are passaged in culture. And what we found was that when these fibroblasts are passaged in culture, that there's a progressive telomere shortening. So what's shown here is a southern blot of human genomic DNA from fibroblasts that have been passaged for different numbers of population doublings. And what you see is this smear that represents all of the telomeres in the cell, and you see this heterogeneity because, as I've told you, in the cell you have continually some shortening and some lengthening, and so telomeres are very heterogeneous in length. But as these fibroblasts are then passaged in culture, you see that there's a decrease in intensity and then also a decrease in length of the telomeres. So this was the first indication that telomere length may play a role in cellular senescence in this fibroblast model. Subsequently, people also were able to identify telomere shortening actually in humans as they age. So this was an example also from Cal Harley. Looking now in total white blood cells at the telomere length, so this would be representing the mean of that distribution of telomeres that you were seeing on that southern blot. And if you look now with donor age over years, what you find is that there's a progressive decline in the telomere length in white blood cells in these donors with years. Now, we know subsequently that these white blood cells are telomerase positive cells, that is, they do express telomerase. And even the CD34 positive stem cell is a telomerase positive cell. And so one of the points that I'm going to be making is that you can have telomere shortening occur even in cells which may have some telomerase. However, if the number of cell divisions is greater than the amount of telomerase to elongate it, you can still see some telomere shortening. So you can have telomere shortening even in the presence of some telomerase. And this will come up as a theme throughout the talk. So we were very interested in the role of telomerase in both cancer and in these models of aging. And so we just decided to use the laboratory mouse to be able to do genetic experiments to test what happens with telomeres. So we set out to look at mouse chromosomes in a mouse model where we eliminated telomerase. So in collaboration with Rhonda Pino, Maria Blasco, when she was a postdoc in the lab, generated a mouse in which one of the two alleles of telomerase was knocked out. So we call this the mouse telomerase RNA, and these are heterozygous mice. And when you cross two heterozygous mice together, you get, of course, the wild type, the heterozygote, and the completely null animal. Now, these animals were born in normal Mendelian ratios, and there was nothing apparently wrong with these mice. And so we recognized that if the telomeres were long enough, there may not be an initial consequence. And so we took these mice, which we call the G1, for the first generation in the absence of telomerase, and we bred them to each other to generate the G2, or the second generation telomerase knockout mouse. We then further bred these, the G3, the G4, the G5, and the G6. And I'll tell you in a minute a little bit about some of the phenotypes that we see in the late generation mice. But of course, the first thing we wanted to know was what's actually happening with the telomeres as this line of mice is breeding. So we used an assay which was developed by Peter Lansdorp at the Terry Fox lab, in which we can specifically measure the telomeric repeats on the ends of the chromosomes. And so this assay uses a small probe that is just 3 telomere repeats, and you hybridize this to metaphase spreads of chromosomes. So what's shown here in DAPI in blue is the chromosomal DNA, and then the telomeres are hybridized, and the signal intensity of each one of these ends is proportional to how long the telomere is. And you'll see in a few slides, the reason that this assay is so nice is that you're actually looking at the telomeric repeat and not all of the subtelomeric variation that may occur if you were to do a southern blot like I showed you for those human telomeres. So when we do this and we quantitate the signal intensity on all of the chromosome ends, when we look in the parents from the cross, what we have here is the signal intensity versus the number of ends that have that intensity. And so you see this nice heterogeneity that really represents that same heterogeneity that I showed you on the southern blot in the earlier slide of the human telomeres. So you see this frequency distribution. But as you look at the G2, the G4, and the G6, you can see that there's clearly a decline in the number of telomere repeats on the ends of the chromosomes, and that the short telomeres are accumulating in these late generation mice. The other thing that's really nice about this assay is that you're actually looking at these metaphase chromosomes, at what's happening to the chromosomes. So in the wild type, which I showed earlier over here, you can see telomeres on all of the chromosome ends, but as you look in first the G2, the G4, and then the G6, you start to see some unusual things. Now, mouse chromosomes look like these little boomerangs. And that's because all mouse chromosomes have a centromere located all the way along one chromosome arm. That is, they're all telocentric. And so you would see the centromere as a constriction up here. Now, in these G6 lymphocytes that we're looking at, when you see chromosomes that look like this, that's clearly 2 chromosomes which have fused through their short arms. And so you can actually see that what's happened here is we've not just lost telomere sequence, but we've lost telomere function, because what the telomere does is to provide stability to the chromosome ends. So as these mice are progressing down increasing numbers of generations, we're actually losing telomere function. So the consequences of the telomere shortening were really quite interesting. And I'll tell you a little bit about the phenotype at the tissue level in a minute, but I'll just tell you that what happens is that you have progressively more cells in the later generation which undergo either cellular senescence or apoptosis programmed cell death. And it's only these late generation mice which show these phenotypes. So what that tells us is that it's not the absence of telomerase per se in these early generations, but rather it's the short telomeres which must be causing the phenotypes. If the absence of telomerase had a direct effect, then you would see that in the first generation. But we don't see any effect in the first generation. And the only difference genetically between this mouse and this mouse is the short telomeres, which have been bred for a number of generations. So that's the first point, is that it's the short telomeres, not the absence of telomerase. And then at the level of tissue, what we see is a number of tissues of high turnover where we see the inability of tissue renewal. So one thing that we can see very clearly early on in these mice is testes atrophy. And so at the 6th generation, the mice are pretty much infertile due to this testes atrophy. So here's a cross-section looking at seminiferous tubules, where you have the cell division around the periphery of the tubule, and then meiosis as the sperm progress inward. And you can see that there's a large number of empty tubules in the G6 mice. Another tissue where we see a large change is intestinal villus atrophy, where you see in the wild type these nice villi, but in the late generation animals you can see regions which are completely absent of any villi. And I'll be telling you about some additional phenotypes that come out a little bit more strongly, such as bone marrow failure and other phenotypes in a few minutes. But what would it be that would cause these phenotypes? We were very curious to know how could it be that simply having short telomeres would cause these dramatic phenotypes in tissues. And I'm not going to have a chance to take you through all of the studies. Our lab has participated in many other labs in the telomere field, have really come up with this model in which the short telomere looks like DNA damage. So normally, when you have double-strand DNA that suffers some sort of a double-strand DNA break, what occurs is that that broken end is recognized by specific proteins, and there's a signal that is sent down a signal transduction pathway that goes through P53, which can signal either apoptosis or senescence. So it turns out that when a telomere is long, it's protected, and this DNA end looks like a telomere, not like a DNA break. However, when the telomere gets to be very short, now it no longer has the protection, and now it looks like a double-strand DNA break. And so there are proteins that recognize this dysfunctional telomere, and there's a signal transduction pathway that also goes through P53 that signals either apoptosis or senescence, probably dependent on the cell type. We're very interested, of course, in what it is that's specifically recognizing the damaged telomere, because we know for DNA damage that there are different proteins that recognize different types of DNA damage. And so we would imagine that there may be some specificity for proteins that may recognize telomere damage. And this is a very active area of research in the field. But nevertheless, that's the mechanism by which we believe through P53 that the cells either undergo apoptosis or senescence. So again, to get back at the question about why does telomerase really matter? I've told you a number of nice stories about telomere shortening and some DNA damage, but how does it play a role in the real world? As I've mentioned, our lab and a number of other labs, including Rhonda Pinno's, has looked for a long time at the role of telomere shortening in cancer. And there is evidence by breeding these telomerase knockout mice to tumor-prone models that in a number of different settings you can actually block the growth of the tumor. But today, rather than telling you the cancer story, I'd like to focus a little bit more on the story of age-related degenerative diseases. So we got interested in this issue of tissue renewal because of a particular finding that telomerase was involved in a human-inherited genetic condition. And the way that this came about was really quite complex, and it shows the nice mixture of real basic science research as it connects to clinical medicine. So in my lab, Junlian Chen, who was a postdoc, was very interested in the structure and function of the telomerase RNA. And he had developed this nice secondary structure model of the human telomerase RNA by looking at phylogenetics and comparative phylogenetics, and then making mutations in these different domains of the RNA. And what he was able to find is that this region here, this pseudoknot template region that has the template that the telomerase uses, binds to the catalytic TERT protein and is needed for catalytic activity. This region here as well binds to TERT and is needed for catalytic activity. And he found this region here, which was a SNO RNA domain. So this is found in many small nucleolar RNAs. And this domain actually had been independently identified by Kathy Collins and her lab at Berkeley. And it turns out these SNO RNA domains bind a complex of proteins, the dyskeratosis complex. So NHP2, NOP10, and dyskerin bind to these particular domains. So this finding was quite remarkable, and it actually prompted Kathy Collins to suggest that maybe dyskerin played a role, that the telomerase RNA might play a role in diseases that dyskerin is associated with. And just the next year, a paper was published by a group in London that were looking at the inheritance of the human autosomal dominant disease dyskeratosis congenita. And so the paper that appeared says the RNA component of telomerase is mutated in autosomal dominant dyskeratosis congenita. So this group was not working on telomerase initially. Rather, they were very interested in mapping genes by taking families and looking at linkage analysis and doing an unbiased search for genes that may be involved in this inherited autosomal dominant disease. And then the fact that the human telomerase RNA was in the region where they had a good LOD score led them to look at the telomerase RNA because of the association with the structure of the SNO RNA domain. And sure enough, in these families, there is the RNA component underlies the inheritance of this autosomal dominant disease. So what are the clinical manifestations of dyskeratosis congenita? The disease gets its name, dyskeratosis, because of skin and nail abnormalities. The skin hyperpigmentation, rashes, abnormal nail growth. However, the mortality of the disease is primarily due to bone marrow failure. There's also an increased incidence of cancer and pulmonary fibrosis. And so we were interested, having spent a lot of time trying to understand the role of telomerase in tissue renewal, about how telomerase plays a role in this particular disease. So I want to just take a minute to go through one of these autosomal dominant families, and this is a family that was seen at Hopkins by Mary Armanios. And you can see the autosomal dominant inheritance pattern here, with the individuals shown in black having the disease dyskeratosis congenita, and the proband that came into the Hopkins Clinic is shown here. I want to make a couple of points about this family as well as other families with autosomal dominant inheritance. And that is that there is genetic anticipation in this disease. So genetic anticipation means that there is a earlier onset and a worsening of phenotypes in the disease. And this is typically thought of in triplet repeat expansion diseases, where you have from one meiosis to the next an expansion of these unstable triplet repeats. But it may also remind you of the telomerase knockout mouse that I was describing to you, where you have a later more severe disease in later generations. The interesting thing was that the affected individuals are heterozygous for mutations in either the HTR, which I showed you from the Docal group, or what Mary Armanios found, heterozygous in the H-TERT mutations, that is, mutations in the protein component. And the fact that the affected individuals are heterozygous tells us that there are two main mechanisms that we should be thinking about molecularly. One is that if you have a dominant negative effect, if you have one mutant allele and one wild type allele, if that mutant allele goes and inactivates the wild type allele, then you can have disease when you're heterozygous. Another mechanism would be if you have one mutant allele and one wild type allele, and simply not having enough of the wild type allele causes disease, and that's called haploinsufficiency. So the mechanisms, either dominant negative or haploinsufficiency, were ways that people were thinking about could play out in this disease. And we realized at the time that we had a mouse where we could directly test whether or not there was haploinsufficiency that causes the disease. And so I'll show you another mouse model that we developed where we were actually able to show that indeed haploinsufficiency can underlie this disease, dyskeratosis congenita. So we were looking at a number of different strains of mice in the lab, and this is again a southern blot looking at a variety of mouse strains. And what's shown here is C57 black mice, and you see this very large smear when you do a southern blot. And so this is the heterogeneity within the standard laboratory strain. However, there were a number of other strains of mice which are recently derived from the wild. So these are all wild-derived mice. And you can see that they have significantly shorter and more homogeneous telomeres. So we focused on one of these, which is called CAST-EI, that has this nice homogeneous telomere length distribution. And interestingly, when you look at a number of different individual mice from various laboratory species, so again, here's a pulse field gel looking at C57 black, 129, FVBN, common laboratory strains, and comparing that to Castaneous, you can see that the Castaneous telomeres are very similar to human telomere length, whereas all of these other mouse strains seem to have lost their telomere length equilibrium and have developed these very long telomeres. So what we did then was to take these CAST-EI mice and to cross our telomerase knockout allele onto this new genetic background where we would have telomere lengths much more similar to the human telomere length distribution so that we would hope to be able to mimic disease. So we had bred the allele then for 6 generations onto this CAST genetic background. And that's when we realized that we had a very good method to look at the question of haploinsufficiency. So what we did was to take our mice now on this new genetic background and take the MTR, cross them to each other as heterozygotes. But instead of following the null allele, now we followed the heterozygous allele. So we followed the heterozygous generation 1, heterozygous generation 2, heterozygous generation 3, etc., never allowing the allele to go completely null. And what we found that indeed at the level of telomere length, there was progressive telomere shortening in the heterozygous state. So what's shown here is the frequency distribution from one of these quantitative fish assays. And so you see in black the distribution of telomere lengths in the wild type Castaneous. The knockout is shown in red, the distribution in the knockout animals. And then the heterozygous generation 1 is shown here in gray, and you can see that that's shorter than the wild type. But more importantly, when we breed down then and now look at the heterozygous generation 5, there's significant telomere shortening. So the shortening from generation 1 to generation 5 represents the haploinsufficiency. So in this case, the mice have telomerase, they just don't have enough telomerase to be able to maintain the telomere length for the number of divisions that these cells have gone through. So, of course, when we're breeding heterozygotes, of course, at each generation, you get out the next generation heterozygote, but you also get the knockouts. So we called this the knockout from the generation 2, the knockout from generation 3, etc. And when we looked to see the survival of these mice, we found that there was a dramatic reduction in survival. So this is now looking from the later generation heterozygotes all the way down, the knockouts that come out at each generation. And you can see the wild type shown here in black, and progressively with each generation, there's a decreased survival simply by breeding these heterozygous mice. And when you actually quantitate that and look at the median survival versus the generations that the mice have been heterozygous, what you find is there's a very nice correlation showing that progressive decrease in survival is tightly associated with the decreased telomere length that's occurring in these mice. We then went on, of course, to look further in the colony and ask the question directly about the survival of the heterozygous mice themselves. And you can see that when we look at the later generation mice, heterozygous 7, 8, 9, and 10, there's a progressive decline in the survival. And this mimics the situation in dyskeratosis congenita, where there's haploinsufficiency and disease. So inheritance of short telomeres decreases the survival of the mice, and it resembles the genetic anticipation seen in dyskeratosis congenita. So the mouse really recapitulates many of the situations that are seen in the human disease. I'll be showing you that there is bone marrow failure, intestinal villus atrophy, chemotherapy intolerance, and genetic anticipation, suggesting that this mouse is a nice model for the human disease. Now, of course, when we're doing this breeding, when you breed hets to hets, you don't just necessarily get hets and knockouts, you also, of course, get the wild type. And this was really the initial surprise that we got when Lingyang Hao, the student doing this breeding, came into my office and said, I wonder what would happen if I looked at the telomere lengths in the wild type mice. And it turns out that when you breed these mice down and now look at the wild types that come out of this line, it turns out they're not really wild types. So we're calling these wild type star. So a wild type 5 star has as its litter mates a heterozygous generation 5 as well as these knockouts. So when you look at the telomere lengths, what you find is that the telomeres aren't restored back to their normal wild type length. So again, here's a telomere length distribution of the wild type telomere lengths and the knockouts just for comparison. I showed you previously that the heterozygous generation 5 is shorter than wild type, but if you look at the wild type 5 star, you can see that these are also very short. I've been making the point that telomerase is limiting. There's not enough telomerase to just completely re-elongate the telomeres within one generation of mice, and so these mice have short telomeres. I've also been telling you that it's the short telomeres that cause the disease in these settings of heterozygous mice. So of course, we wanted to ask then, what about the phenotype in these wild type 5 star mice because they have short telomeres? And sure enough, these mice also have the phenotypes that we see in either the knockouts or the heterozygous mice. So this is just one example. Looking at the aberrant tubules, if you look in wild type and compared to wild type 4 star, which have short telomeres, you can see that there's a significant increase in the empty tubules. And so what we call this is genetic disease in the absence of the mutation, or an occult genetic disease, because you could sequence the locus which caused this disease, in this case the telomerase RNA locus, and it of course would be wild type, and yet it was the thing that caused the disease for generations earlier. And so we think that this is important for a number of reasons. First of all, this is an inherited human disease, and there may be family members where the mutant allele is segregating in the family, and there may be one brother who has bone marrow failure and has the mutant allele, but there may be another brother who doesn't have the mutant allele, but that doesn't mean that he doesn't have short telomeres. There may be some chance that those individuals would have short telomeres. And in fact, there are several people studying dyskeratosis congenita who have evidence that this may actually be occurring, that the telomeres are somewhat shorter in relatives of those that have mutations in telomerase. So this has important implications for the treatment of individuals with these inherited human diseases. But what we realized was that not only does it have implication for families with these diseases, but it also has more widespread implication for individuals with short telomeres who may not come from families with these particular inherited diseases. So if you actually look in the human population, and this was a study published a couple of years ago by the Valdez group, where they did an experiment similar to what I showed you at the beginning of the talk, looking just in total white blood cells at telomere length. If you look at telomere length versus age in the human population, you find in these white blood cells that there is a decline in telomere length with age. But what I want to point out to you here is the wide heterogeneity in the human population of these telomere lengths. And what we realized is that with these wild type star mice, we now had mice that we had created which have very short telomeres but aren't mutant in any way. They are just normal wild type mice because the telomerase allele has been restored. And we thought that this would be a very nice model to try and understand what actually may happen in normal aging in individuals that may have relatively short telomeres in the population. And so we use these wild type star mice then to characterize what one might expect of short telomeres in the aging population. And we found that there are significant defects due to the short telomeres. So if you look at bone marrow cellularity, when you look at the cellularity of wild type and compare that to the wild type 8 star, we see that there's a reduction. And if you look at spleen cellularity, again, there's a significant reduction in the spleen cellularity. And you can compare that to the litter mates, the HGH and the knockouts. There's a significant reduction. When we look and quantitate B cells and T cells, again, there's a reduction in both of these compartments in these wild-type animals with short telomeres. So here looking at B cells, comparing litter mates again, the wild type to the wild type 9 star. And in T cells, the wild type to the wild type 9 star, there's a significant reduction. When we then look a little bit more closely and look at the ratio of the CD4 to the CD8 cells in these mice, what we find is that there's a significant decrease compared to wild type in the CD4-CD8 ratio, and this ratio is really a hallmark of immune senescence. So I just wanted to reiterate that although we've been talking about individuals with mutations in telomerase, the fact that the short telomeres are really what's causing this phenotype would suggest that the components that go into the inheritance of these very variable telomere lengths in humans may play a role in susceptibility to a number of these age-related diseases. Of course, we were very curious since our wild-type star mice didn't have normal telomere length, and we presumed that was because telomerase was limiting. So we wanted to ask the question, can we restore the normal set point of telomere length? And in fact, it took many generations of breeding. In fact, it takes 5 generations of breeding, but we can restore the normal genetically determined set point in these mice. So what we did was to take the mice that had come from a heterozygous cross. These are the HG4 mice. So these are going to be a wild-type 5 star. And now you have to follow me here with the nomenclature. It gets just a little bit hairy. We wanted to cross the 5-star mice to 5-star mice because we didn't want to accidentally bring in extra long chromosomes and make our studies more complicated by crossing to mice that already were bringing in long chromosomes. So what we did was we called this the wild-type 5-star 1. We did this cross, and now we get wild-type 5-star 2, wild-type 5-star 3, etc. And when we follow the telomere lengths, the heterozygous parents, the wild-type parents are shown in this blue distribution, and the starting heterozygote parents are shown in red. When we look at the wild-type 5-star 2 here, we can see that there's been a shift to higher telomere lengths, and now 2 more generations of breeding, wild-type 5-star 4 has restored the genetically determined telomere length set point. Of course, we wanted to look at the phenotypes because I've been arguing that the phenotypes have to do with telomere length. And if you look at the mutant telomeres, the aberrant testes in these mice, and compare wild type to the heterozygous generation 4, where I've shown you there are empty tubules, we have some in the 5-star 1, but as the telomere length is restored, the phenotypes are rescued. So one of the questions that we wanted to follow up on then is in the human disease literature, there are individuals in these families which are segregating telomerase mutations who are mutant in either the RNA component or mutant in the protein component of telomerase. And there's a fair amount of discussion in the literature as to whether you might expect different disease phenotypes for mutations in one component versus the other component. And in fact, there has been a fair amount of interest in the protein component of telomerase, TERT, in papers where people have overexpressed this protein component and found additional effects that are separate from the telomere. And so we wanted to know whether this may actually play a role out in disease. So some of those examples are that the TERT gene may regulate chromatin state and DNA damage response. The TERT is suggested to be both a MYC and a Wnt-related transcription factor, and another paper suggesting that the TERT protein is actually in the signaling in the Wnt signaling transduction pathway. And if this extracurricular role for the protein component of telomerase plays out in humans and in mice, then what you would expect is that there may be different phenotypes that you would have in families that have mutations in the protein component. So we set out then to ask, would we see a difference in the null state for mutations in the RNA component or mutations in the protein component? And what I'll tell you is that we don't find any differences in the phenotype, but the way that we wanted to do this is to look both molecularly as well as at the mouse in an unbiased way. So the first thing that we wanted to do was to ask whether we see any dramatic difference in the transcription profiles when we acutely remove TERT. So we do a heterozygous cross and we look in the embryos that are derived, and we look on microarrays. And these are 3 different biological replicates of looking at the TERT compared to wild type. And what you're looking at here is a volcano plot where the p-value for all of the genes is shown. The significant p-value is in this quadrant. and in this quadrant, and the fold change, that is anything that's twofold changed downward is here and twofold change upward is here. So we didn't find any genes that were significantly changed in their expression level when we looked on these microarrays comparing the protein component knockout to wild type. Also, as a control, we compared the RNA component knockout to wild type, and we didn't find any genes that were significantly changed in expression. Of course, a better way to ask about disease is to actually look in the mice and see if these might be any different. So what we did was to take this allele that is knocked out for the TERT, or the protein component of telomerase, and this was originally from Lee Harrington's group. And what we did was to cross that onto the castaneous background for 7 generations. So now, rather than having these very long heterogeneous telomeres, we have very short homogeneous telomeres, where we can compare the phenotypes that we see in these mice to the mice that I just described to you, which were knocked out for the RNA component. So again, we wanted to ask the question, would the TERT gene also show haploinsufficiency, which was suggested from the human genetics, because the autosomal dominant inheritance is in people that have both the protein component mutant as well as the RNA component mutant. And then we also wanted to characterize the null phenotype to see if there may be any additional phenotypes when TERT is completely absent. So the first thing that we were able to show is that you do have telomere shortening, and the rate of telomere shortening, whether you knock out the protein or the RNA component, is the same. And we can show that there's haploinsufficiency at the level of telomere length. So this is now the TERT knockout. The wild type is in blue, and the TERT knockout is in red. And 2 heterozygous littermates that have come from this cross are shown here, and you can see that they're intermediate. So there is haploinsufficiency for the protein component as well as for the RNA component. We look at the survival curve of these mice, and just as I showed you for the RNA component knockout on this castaneous background, there's a significant decrease in survival with each generation of the heterozygous parents. We were able to collaborate with Bak Karam in the pathology department at Hopkins and do a blinded study of total animal necropsy. So he didn't know the genotypes of the mice that we were giving him. And what he reported back to us on total necropsy was that there were empty seminiferous tubules, intestinal villus atrophy, and evidence of bone marrow failure. And he didn't find any additional phenotypes. We then wanted to explore the question of haploinsufficiency and whether or not you may see some of the phenotypes in these mice which are heterozygous. And again, if we look at the seminous tubule atrophy, we look at the group of the latest generation heterozygous mice, and we find a significant increase in tubule atrophy. And when we look at white blood cells and just the whole blood counts, comparing wild type to heterozygotes, there's a significant change in the white blood cell count, again suggesting that there's haploinsufficiency for the TERT protein. We looked specifically at one pathway which was reported to be involved in the Wnt transcriptional pathway, and that is skeletal development. There was one report that TERT knockout mice may have abnormal numbers of ribs. And so we looked both in our C57 black colony at the rib number, and the normal rib number is 13. And what we found is that there were no abnormal rib numbers either in the black 6 colony or the castaneous colony. So we didn't find any evidence of developmental changes that may affect the skeleton. So what we conclude then is that the phenotype of the MTR or the RNA component knockout or the mTERT, the protein component knockout mice are similar, indicating that it's the short telomeres that are causing these phenotypes, and the phenotypes that we see are very similar. So I just wanted to go back and reiterate some of the studies that we did with our wild-type star mice and point out that many of the diseases that are seen in families with insufficient telomerase share features of age-related diseases. So bone marrow failure, I talked a little bit about immune senescence, chemotherapy intolerance. I haven't had a chance to talk anything about pulmonary fibrosis, but that's also occurring in these families, liver disease, and increased incidence of cancer. And these are all in addition to segregating in these families, are examples of a specific type of age-related disease, and these have to do with diseases that have to do with cell turnover. So the combination of the initial telomere length, where you may be on that wide distribution of telomere lengths in the population, and cell turnover in vivo may predispose to age-related disease. So if you think about in the immune system, we know that when cells die, other cells have to divide to replenish them, and so those individuals that may have gone through more rounds of cell division may have somewhat shorter telomeres in that distribution than individuals that didn't, indicating that it's both genetic and environmental components which make up that large heterogeneous distribution of telomere lengths in the human population. So just to summarize what I've told you, I told you that telomeres are required for chromosome end protection. Telomerase is essential for telomere maintenance. Telomere shortening leads to cell death or senescence after many cell divisions. Short telomeres limit tissue renewal. Loss of the RNA component and the protein component result in the same phenotype. Telomere length may predict the onset of certain kinds of age-related disease. And just to give credit to all of the people who did this work in the lab, the telomerase knockout mice were looked at by Sophia, as well as the TERT knockout mice together with Margaret Strong. And our collaborators in pathology, Bach Karam and David Huso, have been invaluable collaborators, and many of the work was done in collaboration with Mary Armanios in the oncology department. So thanks.
So thank you, Carol, for a very interesting and clearly expressed set of experiments that teach us a lot about this interesting system. We're now open for questions, so there are microphones at the aisles. Please, if you have a question, migrate to that spot and perhaps identify yourself, and Carol would be glad to address you. Go ahead.
Hi, I'm Orna from NIDDK. You mentioned sort of in passing that the patients with dyskeratosis congenita and also towards the end have cancer or increased incidence of cancer. I guess that would be surprising given that you need telomerase for proliferation. So could you elaborate on that?
Yeah, this is actually very interesting. So what we know is that probably 99.9% of the time what happens when the telomeres get short is the cells either die or undergo senescence. However, it may be that the cells that escape that can go on and initiate tumor formation. The interesting observation is that in the human population, the people who primarily have bone marrow failure get leukemia, but the people who primarily have this disease, pulmonary fibrosis, get lung disease. So it's not just that they are in general cancer prone, but they get the specific cancers in the same tissues where they're clearly having the stem cell failure. So we find that very interesting, and it's not clear whether it's genomic instability, initiation of genomic instability, or some sort of tissue remodeling which may initiate that tumor formation. But this is clearly seen in the human patients, and we can see microadenomas in our mice. So in the intestines, in the regions where we see villi atrophy, we also see microadenomas, but we haven't seen those actually progress to frank cancers, possibly because they don't have.
So if you look at their karyotype, do they have typical chromosomal abnormalities, say chromosome fusions and things like that, or it's anything?
We haven't been able to address that in the mice. I think it's very interesting because we now have a mouse model that is heterozygous, and the humans are heterozygous. So it's not like they have no telomerase. They get the short telomeres because they're heterozygous. So it's somewhat different than what people have talked about, where there's pathways by which telomeres can be maintained in the absence of telomeres. Here, we would have telomerase. So one question is it upregulated? But the gene is still there and it's on.
The question relates to the restoration of the telomere length. So what happens during the induction of pluripotent stem cell? It happens in generations that you could restore. So what happens to the telomere of a young person versus elderly person in terms of the telomere length of the iPS cell?
Yeah, I think that's interesting, and there's a number of papers that have been trying to look at the question of can you restore telomere length in iPS cells? And I think that part of these are going to have to do with the protocol and how long you grow cells. We know under normal situations that any given cell cycle, telomerase only elongates a couple of telomeres. In fact, it elongates the shortest telomeres, which probably is the reason that it takes us 4 generations of mice to restore back up to the wild-type set point. And so I think that this is something that will be of interest, and just transiently restoring telomerase activity may not be enough to restore back to the appropriate telomere length. So I think that this is an interesting question.
But you also showed the association with c-MYC, and c-MYC is one of the transcription factor used.
That was a paper that suggested that overexpression of the protein component of telomerase played a role in c-MYC. We don't find any evidence for changes in c-MYC when you knock out the telomerase. So my suggestion would be that may be a neomorph in which the overexpression is interacting with additional pathways that the telomerase doesn't normally interact with.
Carol, can you restore telomere length by overexpressing telomerase, for example, under a tetracycline-regulated promoter in a generation like generation 5, where you've crossed heterozygotes? Have
you been reading the New York Times recently?
Well,
yeah. So Ron DePinto just published that 2 days ago. So what he did was, yeah. So
the answer, you can do it.
Yes. So he bred down a Cre-ER null. For 4 generations, and then turned it on and found that you could rescue a number of these phenotypes that I've been talking about in the intestine and a number of other phenotypes. And we did that by breeding down for 6th generation and then crossing to a heterozygote. And in the next generation, all the phenotypes are restored. But it's a little bit nicer trick to do it with Cre.
So Carol, you showed a couple of times diagrams that reflected telomere length in circulating white cells as a function of age. Clearly there is a relationship, but it's a blurry one with a lot of scatter. If we were to measure the telomere lengths of everybody here and plot that on a diagram, could you predict longevity in all the people who are listening to your lecture?
Clearly, one of the points I was making about the heterogeneity is that you can't really predict. You couldn't take someone's white blood cell and say how old they are even. People have said, can you use this to say how old they are? However, it's very clear that in the clinical disease, Peter Landstorp has a very nice way of measuring where you are in the distribution. And those individuals that fall below the 1st percentile are highly predictive, like 95% that they have mutations in the family. So yes, those individuals that are way off are highly at risk for a number of these diseases. So you can predict disease risk based on where you are in the percentile in the distribution.
Have life insurance companies started to ask for this information?
Isn't there something called GINA that I've heard a little bit about?
Life insurance, I'm sorry to say, so don't get this yet. Well, okay, if there are no other questions, we're going to have refreshments, actually not in the library, but back here. You're all most welcome to come and continue the conversation with the speaker. Please let's thank Dr. Greider again.